Concept 16.2, DNA Replication and Repair. In this concept, we will be discussing semi-conserved replication, DNA replication, proofreading and repairing, and the ends of linear molecules. So, we now know that what the structure of the DNA molecule is. The next thing researchers needed to figure out is how it is replicated. They came up with three possible models, or three hypotheses. As shown in this figure, one hypothesis is that a completely new double helix was constructed from the original double helix. See, the light blue lines represent new DNA strands. This is called the conservative model, or the original double helix is conserved or unchanged. The next hypothesis is that the original strand is split and a new strand is built on the separated original strands. See, we have a light blue strand and a dark blue strand in the mix for the first replication. Then light blue with dark blue for half of the next round, with all light blue for the other half. This is called the semi-conservative model. The last model, the dispersive model, has the original strand mixed with the new strand as shown here. You can read about the experiment that provided evidence for this semi-conservative model. It was a very well-designed experiment. I wish I'd come up with something that elegant myself. What I want you to remember, though, is that the semi-conservative model won. Now, let's take a look at how semi-conservative DNA replication works. Several enzymes all come together to replicate the DNA during S phase. Let's start with helicase. This enzyme settles down on the double-stranded DNA strand on a special spot called the origin of replication. It is, to use a human example, the sign on the board game that says "Start here." Once helicase binds to the origin of replication, it unwinds the helix, that's why we call it helicase, and breaks the hydrogen bonds between the bases. I call this unzipping the helix because you end up with two single strands that look like an unzipped zipper. Now those unbound bases want to get back with their complementary bases. So proteins bind to the single strands to keep them single. We call them single-strand binding proteins. Don't you love it when the name tells you what it does? As helicase unwinds the helix, it gets wound tighter and tighter in front of the replication fork. See, this here is called the replication fork. This is called supercoiling. Topoisomerase is an enzyme that unwinds this supercoiling. There is a type of RNA polymerase, or an enzyme that makes a polymer of RNA, you may want to review Chapter 5 for this part, called primase, which makes a short strand of RNA that is complementary to the template strand. We call this strand of RNA a primer. Primase does this because DNA polymerase III can't bind to a single strand of DNA. Think of single-stranded DNA being a 1/16 inch nut, and DNA polymerase III is a 1/8 inch wrench. The active site is too big for the single strand, and the single strand rattles around in the active site. The primer makes the strand double-stranded, so DNA polymerase III can make a new strand of DNA using the old strand as a template. So if the old strand reads 8AT, the new strand will read TTA. That way, you end up with two new strands that are the same as the original strand. Let me point out here that DNA is always synthesized in the 5' to 3' direction. That's what this means here. So this strand is being synthesized continuously in the direction the DNA is being unwound, or the direction the replication fork is opening up. We call this the leading strand. So what about the other strand? The other strand is the lagging strand. Is still being synthesized in the 5' to 3' direction, but it is going away from the replication fork. DNA polymerase III keeps running into the primers because primase is continually making new primers behind it. So the lagging strand is being synthesized in short fragments called Okazaki fragments after the researcher who discovered them. Eventually, all of these RNA primers, both in the lagging strand and in the leading strand, need to be removed and the various pieces hooked together. DNA polymerase I, another type of DNA polymerase, replaces the RNA primers with DNA nucleotides. Then DNA ligase comes in and hooks the various pieces together. So you have one long continuous double-stranded DNA molecule. I have a lovely animation that goes all of this. Here's the website to give credit where credit is due.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides, complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Hopefully that animation helped you visualize what's actually going on. Feel free to watch it more than once. So, no enzyme is perfect, and that includes DNA polymerase I. Sometimes a T nucleotide gets in where a C is needed or vice versa. DNA polymerase III proofreads what it has just done, so if there is a mismatch, it takes it out and puts the correct nucleotide in. But sometimes, just like with people proofreading their papers, DNA polymerase III doesn't catch all its mistakes. This causes a bump in the double strand, like we see here. Mismatch repair enzymes follow the DNA polymerases to look, so to speak, for bumps and put the correct nucleotides in. That takes care of errors during replication. What about damage that occurs after replication and during G1, or the cell's normal life? Radiation and certain chemicals can damage DNA. Other enzymes, called nucleotide excision repair enzymes, take the bump out of one side and put in the correct nucleotides. Linear DNA, such as that which is found in eukaryotic cells, have a problem. The 5' ends are not synthesized because DNA polymerase can't go 3' to 5'. This could lead to the chromosomes becoming shorter and shorter with each S phase until important genes are damaged. Well, eukaryotic cells have a couple of solutions. One is a stretch of junk DNA at the ends called telomeres. Actually, telomeres are repeating sequences that don't contain genes, so if they are clipped, no harm. In differentiated cells that have a limited lifespan, the shortening of the telomeres keeps track of how many times the cell has divided, keeping track of how old the cells are. Enzymes called telomerases can lengthen the telomeres to keep non-differentiated cells immortal. Well, that's it for this concept. Make sure you come to lecture with any questions you may have.